Okay, good morning again, and welcome to Lecture 8. So in today's lecture, we will discuss virus, like the basics and the first introductory of your virus. And as an advertisement for spring, and we have scheduled a virology or concept of virology Bio 411 for spring. So if someone's interested in learning more about virus, and you can look forward to that class. That's a 2-hour, 2-credit lecture only. And we're trying to get researchers, virologists from Albert Einstein to teach the class for us. Okay. So we'll cover the basic structure of the virus, and then we will use 2 bacterial virus as an example to talk about 2 types of virus, lytic and more complicated, what's called temperate virus called Lambda. And then we will discuss the vertebrae virus, which will include, at the end, I'll mention the COVID-19. And we will have another lecture, spend more time on actual COVID-19 and how it works. But today's lecture is to lay the foundation for all the virus, right? So the basic, to understand the virus, the basics of virus, we need to understand the structure and its genome. And at the very basic part, a virus has there are 2 basic types. One is the what's called the naked virus, and one is called the enveloped virus. The difference is that the naked virus has 2 parts. It's called the capsid, which is the shell. We call it capsid, all right? And it's made of protein. And then what's inside is nucleic acid. When you see the word nucleic acid, this means it could be any forms of DNA or RNA. Get to that in a second. Okay, so so basically you have genetic material or genetic information protected by a protein shell called capsid. So that's the basic form. And then you have what's called the enveloped virus. The enveloped virus have a little bit more complexity, and this is has a envelope, and it's a membrane. It's a membrane envelope, right? And this is more common among the eukaryote virus for like humans or animals. And so if we go back to the capsid is a very symmetrical structure. And so if you were to look at this capsid, it's very you see it has these points, and then it has the symmetrical structures, okay? And and the one of this the important part is that you see these are almost like a triangle. You see, it's triangle, triangle, triangle. Everything is repeated. So this pointing protein is repeated so many times, right? Here, here, and here. And then in between, these are different proteins. And the main advantage is that it's assembled into a triangle by a few proteins. So at minimum, you need 2 or 3 proteins, and that's what usually happens. So the virus is, because it's small, the genome is a lot smaller. And when the genome is smaller, it wants to code, the numbers of genes gets reduced that it can hold. right? Just think about your textbook that you have, probably have 30 chapters, right? So it covers a lot. But if you limit the thickness of your textbook to, say, 10 pages, then the numbers of characters or the numbers of chapter will have to be greatly reduced, right? The same concept here. So the viral genome is very small, and it encodes a very minimal amount of genes. So the numbers of protein need to be reduced. And then what they ended up doing is that they have these proteins that forms a structure, and then a structure gets repeated. And so that's why you have these isopedric structure. Yes, the genome needs to be small so it can fit into this capsule. We'll come back to this concept later. So this is one type of virus. And then there's a virus called linear virus. And this is an example of linear RNA virus. There are linear DNA virus as well. And so what's unique about this virus is that its genome is coded by one protein. You see, these are the proteins. These proteins are the same. It's almost like your shoe, right? It just fits in there, then goes around the DNA, the RNA, and then it rotates. So all the virus need is just one protein in theory, but in fact, there's other proteins that it makes at the beginning and the end. Okay. Now, just to add a little bit interesting history is that there's a virus called bacterial virus called M13. M13 is linear DNA virus, and it's the first cloning vector. Okay, so when you think about cloning, is putting a gene into an organism, right? The first experiment was putting a foreign gene into E. coli. And this M13 is one of the first bacterial virus, linear virus that's used. Because it's a linear, it doesn't really care how long it is. As long as you have these DNA gets coded, right, covered by the protein. And so, sorry, I need to, okay, thank you. I need to click, now I understand. So the M13 is the first cloning vector, and when it gets, so it's a linear, and so if you put a foreign gene in there, it just gets longer. And so it doesn't really matter. And that was the first experiment that they were they were able to insert a gene. So now this is, but it's very difficult to use. And later on, we migrated to use plasmid, and that's much more easier to use. And so that's a little bit of history, interesting. So if we were to look at the viral genome, this is really just to show you that it could be anything. So you can have DNA, RNA, RNA to DNA, DNA to RNA. All right. And then in between, in more detail, is that you can have single strand SS. So that means single strand. Okay. SS means single strand, and DS means double strand. Single strand DNA, double strand DNA, single strand RNA, double strand RNA, and single strand RNA or double strand RNA. These are all the possibilities. All right. So now you understand the virus, the basic structures has a protein protects the nucleic acid, and the nucleic acid could be a variety or anyone carry any different form. And the structure of the viral protein capsid is very, very simple because the genome is small. It could only code for certain small numbers of genes. So they use a small number of genes to make a repeating structure so that they can achieve and to put together a spherical structure. And then you have an even simpler form of the linear virus, right? And so those are the basic types. And then the one with the animal virus gets a little bit more complicated because they can have an envelope, all right? And that envelope usually is a membrane protein, a membrane that's embedded with lots of proteins, and that adds to the to protect the virus and actually enhance the virus that getting into the host cell, especially the eukaryotic cell. Okay, so now what we're going to do, so those are the basic structure of virus, and now I want to use one particular bacterial virus called T4 as an example to talk about the life cycle. Okay, T4. And so there are T4 and there are T2, 2, 4. And then, so now in terms of phage, so we often use the word phage to describe bacterial virus. Okay, so if I were to correct this, it would be bacterial virus phage T4. Okay, so for the T4 or for phage, the life cycle starts by attaching itself to the surface of bacteria and then getting into it and then reproduce and then come out. All right, and so these are the life cycles, and so I'll go through them individually, and then in the end, and I'll talk about how you measure the growth of phage and what's the application. That's the transduction. Last lecture we rushed through to say, okay, transduction is to bring DNA from the donor to the recipient by a phage. And so we defer that to this lecture. Okay, so we're going to start with T4. And T4 is actually slightly more complicated. And this is an EM picture of T4. And this is on chapter 9, the figure 9.5. So if you look at this, then you have a head, you have a tail, you have tail plate, right? And then you have tail fibers. Okay. And so if we were to look at the life cycle, it goes through these different stages. So first it attaches, and then it gets, the important part is the genomic DNA, the virus DNA gets into it. Okay. Now the DNA injects into the host cell, and the whole now the foreign DNA gets into the cell, penetrates, injection, and then the DNA, the viral DNA is made in such a way that the host bacteria does, it's harder for them to distinguish. They disguise them as a bacterial DNA, trying to hide and get injected, right? Injects into it. And then if they pass the defense of the bacteria, now we know there are different ways of a bacteria to defend these, to destroy the foreign DNA gets into it. So you have CRISPR, right? That's now we know it's that's a defense mechanism, and you also have the restriction enzyme. So bacteria have multiple ways of really cutting off foreign DNAs. Now, but if the foreign DNA is not cut off, and then what will happen is then the foreign DNA will be using as a template for transcription and translation. Now you have proteins, and the proteins and the DNA gets replicated. The protein gets synthesized now, and these self-assemble, and then the last stage, it releases. Okay, so these are the general life cycles that all bacterial virus goes through. So we're going to need to talk about the individual stages more closely. So in terms of attachment, okay, so attaching the virus, the T4 virus, uses the tail fiber to attach to bacteria surface. Now you recognize this is a gram-positive bacteria or gram-negative bacteria. Now that was drawn here. Anyone?
This is a gram-negative.
Ah, yes. Thank you. And so the reason that this obviously is gram-negative is that you have outer membrane, you have polysaccharides, and then you have proteins. And the binding of this virus, the phage, to the bacterial surface is very precise and very strong. And they have to be very specific. So they will land and they will really bind, almost like a train, right? We know the subway or the train, you get two of them together, right? They hook and then they stay together. Okay. Now, the binding is so specific that the bacteria virus that attacks E. coli will only attack E. coli because they have specific surface protein for the virus to attach. Okay, so that has applications. So sometimes you can think about using, if you have a specific bacteria. So for example, Pseudomonas is a bacteria often infects patients. If you have a burn patient, like if you have you burn the skin accidentally, and you may have bacterial infection, and those bacteria tend to be Pseudomonas. And one of the therapy being explored is to spray virus that specifically to the that bacteria, Pseudomonas, and so to kill them. And so the point there is the tail fiber and the protein or the receptor on the surface is binding is very, very specific. And once it binds, then the DNA, T4 DNA, gets injected into the bacteria. Okay. So then the question that I want to throw out to you is that in terms of binding, so the phage binds to E. coli and kills it. And then if the E. coli don't have the phage receptor, sometimes the bacteria stop making the receptor at certain stage. So for example, at stationary phase, they will actually stop making a phage receptor, and then they won't be able to. Then they will be resistant to the phage. They cannot be killed. So then the question to you or to the class is, well, since bacteria is capable of stopping making the phage receptor and be immune to the phage infection, why don't they stop making the phage receptor all the time so that they don't ever get killed by the phage? Shao, go ahead.
I kind of have two ideas clunking around in my head. Either A, it's an extremely energy intensive process to not make the receptor, or B, the receptor is necessary for like growth and development of the bacteria.
Which one do you think is more likely? If you have to rank your choices.
Gun to my head, it's probably growth and development, and then energy intensive.
Yeah. Oh, well, energy, bacteria wouldn't produce anything energy intensive just for the purpose of providing a landing site for the virus, right? And so, no, it's essential because the bacteria is actually need those protein for those growth period. So it's an essential protein, and the virus obviously, or the phage obviously took advantage of the fact that they had to produce this. You can't get away. I'm just going to grab this, right? So that's the receptor in the story. Okay. So now we're going to look at what happens when DNA gets into the host. Now, the first thing I wanted to point out is how long the phage is inside. Once it goes in, once it comes out. 25 minutes. 25 minutes, right? And think about if if I sent you to shop, to grocery shopping, and I give you 10 minutes or maybe 25 minutes to shop for the whole week, and I'm going to leave. And so what would you do? You're probably going to plan ahead before you're getting into the store and say, okay, I know this place. Well, obviously, you go to a store you know very well because now you're limited. And two is that you probably have to plan, okay, I'm going to get these 20 items and I'm going to go from one end to the other end, and then I'm going to go very, very quickly, right? Because the store is big. Maybe I'll send you to Walmart, even bigger, right? So you may have to push your car, you run around, you know the spot. And the point is that because the virus is, the phage has a very limited time, they want to be fast. And everything is very well timed and sequenced. Okay, so so let's take a look at how the T4 virus actually programs itself so that it can complete its life cycle in 25 minutes and get out. Okay, so we're gonna break the life cycle in two parts: the early part and the later half. So the early part, and so this is the early part. So now the injection of the DNA, right? So infection starts at 0, and what happens here in the infection? It's the DNA gets in, right? It didn't show. The DNA gets in, and then the next thing that needs to be happen is gets transcribed. DNA, if it's avoided the digestion, which E. coli will attack, or the bacteria host will attack, but if it survived, what will happen is that it will get transcribed to make early mRNA. Now, we know when the DNA gets into it, and what is the protein does the transcription? Anyone? What protein does the transcription? If you have to think about it, that's a sign that you need to review the previous lecture, right? And so transcription is done by transcription. There's no RNA polymerase 3. Transcription is done by RNA polymerase, right? And the RNA polymerase has two parts: the holo enzyme, or the core enzyme, and the sigma factor. Now, what does sigma factor bind in order to transcribe? It's the promoter, right? So, the early mRNA needs to be transcribed by the host's RNA polymerase and sigma factor, the host, the E. coli sigma factor, right? Which means the virus, well, the virus DNA will actually have a bacteria promoter for its early mRNA. So it can be transcribed by the host. So, and so the promoter here, so the early promoter and that's an issue, right? And so that promoter needs to be similar to the host. So that could be recognized by the host sigma factor. Now, now the transcription takes place. Once it's transcribed, and the mRNA will be translated into protein. Now, what protein or what machinery does the translation? tRNA carries the amino acid, and so what protein or protein or protein complex does the Lewis? Thank you, Lewis. Ribosome does the translation, and that's the bacteria, the host's ribosome. Okay, so and so now it's translated. So now you have the early protein. Okay, so what are the early protein? Well, the early protein mainly have these three type of enzyme. Nuclease. What's nuclease? Anyone want to guess? Nuclease is the enzyme that cuts the DNA, right? That's nuclease. And so the enzyme, so now these nuclease is digesting the host's DNA. Why do they do that? That's because they need the nucleotide to make phage DNA, right? So to produce, to make phage DNA, they need DNA polymerase. And so the phage DNA polymerase So the nuclease cuts the host chromosome into pieces. And then so produce lots of nucleotide. Just think about if you want to build a new house very quickly without buying new bricks. And if you have an old house, and what you do, you just knock down the old wall and then take out the brick and then build a new house, right? And without waiting for the manufacturer to make new brick, and because that takes time. And so the DNA polymerase then make phage DNA. In addition, they also produce new sigma factors, because at this point, what they want is really to make more phage-specific, to transcribe phage-specific genes, right? And so now what happened is now you have the phage DNA, and then you have the new sigma factor. And so now the middle mRNA gets transcribed and then translated. Okay. And so now let's take a look at the DNA replication itself, the phage DNA replication, which is an interesting process. And so what they do is the T4 will, yes, early protein includes the nuclease that cuts the host chromosome DNA into nucleotide so that they can use the nucleotide to synthesize phage DNA. And phage DNA, so the phage DNA is synthesized into pieces. And then these staggered or cohesive ends will stagger together so that they can actually make a long DNA molecule and then cut into the precise size. We'll come back to this in a second. It's a uniform size. They make a long one and they cut it into the uniform size. And the phage DNA polymerase is actually different. So this has two proteins. This is T4 DNA polymerase, and the T4 DNA polymerase is very, very fast. Now you understand the speed, right? And so once the DNA is made, medium protein is made, and then it's the late mRNA gets transcribed and translated, and these late proteins are mainly the shell. And these proteins are also very interesting that these proteins are self-assembled. The phage head is self-assembled. So meaning they will stick to each other. So if you put an A, you produce lots of A, and then protein A, and then you expect the protein A to be sticking to protein B, right? So if you want to make A, B, and then you make A first, then you make B second, and now you have A and B mixed together, and they will bump into each other and form A and B complex, right? And then if you want to put the C next, then you put A, B, and then you produce C. So now A and B already form a complex, and the AB complex will run into C and then forms a ABC complex, right? And so the sequence of the protein that's made resembles, it's exactly the same sequence of they are being assembled. So for example, the tail protein gets made first, and the head gets made first, and then they will make these tail protein. And then the last thing they will make is the tail fiber. Okay. At this stage, when they have the phage is made, and then you have phage head, and then the DNA, the phage DNA, remember it's in uniform pieces. This is uniform pieces, right? And then the phage DNA gets packed into the phage head. Okay. Now the phage head, the phage, since they can't really check which DNA is what, right? They go by the size. If it's the right size, then they know it's the phage DNA because they want to pack the fully assembled, complete phage genome. If it's too long, too short, they're not phage DNA, or it's not ready to be packed. And so once it's packed and then it's sealed by protein here, and then the phage tail is attached, tail fiber is attached. Okay, all these are happening inside the bacteria cell. So think about the bacteria. Remember, the bacteria is really like a water bottle. Everything is floating around, and they rely on assembly and bumping into each other to assemble, right? And so, so that they had to make it in sequence and run into each other very efficiently. They bump into each other, they fit, and miraculously, you actually produce phage, mature phage. And so in this process, it's an assembly process. And once it's assembled, then you have an active T4 lysosome is being produced and being activated, right? Because you don't want to activate the lysosome too early, because if you activate that, and these phage parts will flow out, and then you can't assemble, right? And so it's so amazingly timed. So at 25 minutes, the whole cell gets lysed, and the phage particle gets released. All right. And so this is the 25 minutes life cycle of the T4 virus. Okay, goes in, comes out, and you can certainly measure it, right? And you can measure if you measure the numbers of phage. So this is the virus count. If you count the virus, this is what you see. There will be a latent period, and then after the latent period, the virus start being released. I mean, this is the lyse. This is the period that the virus, the whole cell lysed and the phage released. And this is a one-step growth, because you go in, you come out. And if you were to track timing it, and you can actually see that the latent period is where the early enzymes made, nucleotides made, protein codes made, and this is the assembling period time. And then it gets released. Okay? So that's the life cycle. But the question is, well, okay, you said, we said, well, you can count the virus. But how do you actually count the virus? Can you actually see it? Yes, you can see it, right? We showed a picture of the virus using T transmission electron microscope, but obviously it will be hard to do these, count the numbers of them each time you have to count them, right? Count the viral particle. So the way we figure out how to, the virologists figure out how to count the virus is to take advantage of the fact that the virus actually killed bacteria and lice. So what you do is the phage, okay? So this is the phage dilution. So you have phage suspension here, and then you have molten agar. Molten agar meaning it's well, we usually typically use 0.3%, 0.5%. I'm sorry, soft agar. This is also called soft agar. That means 0.5% agar. Okay, and I think I can move, right? Can I? Well, anyhow, so molten agar equals can also be called soft agar, and which means we use 0.5% of agar in the concentration. And so what happened when you use less agar, and it will still remain liquid at, say, 50 degree, and it wouldn't 50 degree wouldn't kill bacteria, would not kill the phage. More importantly, it will not kill the bacteria. And so then what you do is you add the phage, you add the bacteria, and then you mix them together, and then you pour on top of a nutrient plate. And the nutrient goes up, and so now now what happen is that you then incubate. It's actually mixed, right? And so now when you grow them, and you actually see a clear area, okay, which is called phage plaque. Okay, these called phage plaque. And so here is a real picture of phage plaque. Okay, so why can you pour phage directly onto it? Well, because you want it to be mixed. You want the phage and the bacteria to mix well. The reason is then because when you want the phage to stay in one place. You want to hold the relative place of phage and bacteria. You want them to stay in one place. The reason is then you can, if you have one phage lands in here, let's say one phage lands in here, right? This is one plaque. And that means a phage, one original phage lands in there and then killed one bacteria. And then it killed the next bacteria. And then it kills the next bacteria, right? And it keeps going on and on until such time that the bacteria stop making the receptor and the phage plaque stop getting bigger. All right. And so each plaque here, these white spot or transparent spot represents one phage. Okay, this is almost the reverse of counting bacteria colony, right? The concept of counting bacteria colonies is put the bacteria colony, put the bacteria suspension on plate, and individual bacteria can reproduce and produce a pile of bacteria, right? And that pile of bacteria become visible. And so in terms of phage, to visualize the presence of one phage, you do this, the soft agar, you do the phage plaque count. And so that's how you count the phage. Question? Okay, so by doing phage count, we now can go and you can then go back and say, all right, we can actually do the phage count, and we can see the how many phages. Now you still can do the well, you can do one step, right? You can sample the phage at different times and then count the numbers of phage, and that's how you get them. Okay, all right. Any questions about this? The life cycles of phage. Okay, hope you are impressed by how efficient the phage is going at this. And they will take all available nutrients from the bacteria and then convert them into the phage particle and grow. So the amount of the phage actually, so this is actually fixed, for bacteria, right? So the maximum amount of phage that a host bacteria can produce has an upper limit, is fixed, because it only has this much nucleotide, right? 25 minutes doesn't have enough time to make more nucleotide. So that's the limitation. Okay, so now we're going to move to a different topic, the generalized transduction. So now, last time we said the transduction is the phage takes one piece of DNA from a donor and puts into a recipient. How does it actually happen? Okay, so now we understand the life cycle of a bacteria, of a phage. Now we can go back and study them. So at the beginning, you get a phage, injects its DNA, and that's the red, okay? The red is a phage DNA, and goes in, and what happened? The phage DNA gets replicated, right? And the host DNA gets cut into pieces, right? Okay, so if I give you a long string, and if you take a scissor to cut it, right? What's going to happen? And you go, let's say I'm going to be very fast. I give you a long string. I get 5 of you to start cutting it. Use scissors, just cut to tiny pieces, right? So what will happen is you take on and then you start cutting. Now, because there were 5 of you and you're cutting, and you know, a circle, you start thinking about cutting, and from the outside and in a random cut. The net result is that if I stopped you in the middle of cutting, right, the resulting string, you can line them up. You have the longer piece, shorter piece, and tiny pieces, right? Because you're cutting it, right? And now, statistically, because you're cutting them, one of them or a few of them happen to be a particular size, right? Because you line them up, you can find almost any size, right? It's a linear process, right? So you can find any size. You will be able to find one particular size happen to be the same size as the phage DNA, right? That's the important part. That's the critical part. Now, it doesn't matter which part of the DNA came from, right? It's a random cut. So if you think about the phage DNA is about 100, a T4 is about 100. Let's say 100, not T4. Let's say a virus, a phage is 100 kb in size, and you should be able to find DNA molecule happen to be 100 kb, because that's you're cutting it down from thousands kilobases, right? And so now that same size of the phage DNA, when it will bump into a phage head, it will just go into the phage head, and it happens to fit perfectly. Well, because it's the same size as the phage DNA, right? And now the phage gets confused. Well, it doesn't, it fits, okay. It just closes it. Now, so statistically, you will always have a frequency of host chromosome DNA gets packed into a phage head, right? Okay, so now when the phage lysosome takes place and the host cell lysed, and then you will have normal phage. Here, say normal phage, and then you have what's called the transducing particle. It's a phage head contains the donor DNA or the bacterial chromosome DNA, right? So it's a mixture. The frequency varies, depends on the combination of bacteria and the host. And so maybe you have 1 in 1,000, or maybe you have 1 in 10,000, or maybe 1 in 100,000. But there's a frequency. And then what happens is that if you dilute your phage particle, and because we know we can count, right? And if you count the phage, and you know you have 1 million phage particle, and then you mix 1 million phage with 1 million bacteria cell. That's easy, right? You can count the numbers. And you match them 1 to 1. That's the critical part. You need to match them 1 to 1. Okay, let me type in here. Match 1 to 1. Okay. And so you match bacteria and phage 1 to 1. Now, meaning each bacteriophage will find a phage will find 1 bacteria statistically, and then after that, and then you wash and you clean them up, and so now this 1 phage particle, the normal phage will go and bind to a bacteria and kills it, and then but a phage or transducing particle will find the bacteria and then put the genomic DNA in. Now, instead, well, because it only carries the donor DNA, right? And so it gets in, the donor DNA gets in, and the donor DNA gets incorporated because it's donor DNA. It's literally the same. So it will not be rejected. So at this stage is the same as the transformation, right? The DNA gets in, and it gets incorporated into the or doing the strain exchange to crossing, right? The two crossing to recombination event that allows, because this is a linear DNA. Remember last time I draw that you have. Maybe I could do it here too again. So if this is chromosome, and I can change, I can change the color to DNA here, right? So, and then if I change another color, let's say green, and if you do a recombination event here, and another recombination event here, so 2 recombinations, and then the net result is what? Blue. So the net result is this. Your donor DNA now is in the recipient chromosome. Oh, maybe I shouldn't use the red because the phage, but you get the point, right? Any questions? The important part is, do we know this piece of DNA, where does it come from? We don't, and it's random. So that's any part of the donor chromosome can be transduced into the recipient. And that's why this is called generalized transduction. Okay. Now, this is the life cycle of the T4 and its application and the generalized transduction. So now I wanted to add a little bit of complexity to the life cycle of Lambda. And this is a picture of Lambda. And the Lambda has no tail fiber. It just uses the tail protein at the end here to attach the host cell. Okay. The life cycle of the Lambda is a bit more complicated. It has 2 options. Okay. So what happens is the Lambda has 2 capabilities. One, it's going through the normal the lytic cycle. So you see, follow the red line and it goes in and kills the host and then comes out. That's it. However, the Lambda virus also have the ability to follow a lysogenic cycle. The green, let's say. It can do this. Okay. Okay, so why does it do that? Now, if you come to Lehman, if we were to lecture in person, and if you have to come to, you go to lab, right? So if you go to lab, you can either walk to the lab, right? Walk to Lehman College in the lab, or you get on a Uber, right? Assuming we pay for it. And so you get on the Uber, and why do you get on the Uber? Well, it's easier. It takes you over, right? You don't have to do anything. Saves you energy. Just go. And then, but if you get on the Uber and the guy said, "Where are you going?" And says, "Oh, I'm going to Lehman College. Can you take me to Lehman College?" And the guy smells and says, "Oh, of course." Yeah, your text question just flashes, and I assume you say the virus doesn't come out, and the virus always comes out. If it doesn't come out, it's dead, and then that virus is just dead. And if you have a mutation of a virus or phage that doesn't come out the host cell, it's dead. Now, is it important? It's not that important because their mutations can kill a phage, but there's billions of phage. They go play by the numbers, so it doesn't really matter. And that's why the viral protein or viral DNA polymerase goes fast, and then it's more error prone. Okay, makes more mistake. They don't check. Okay, so let me go back to the lysogenic pathway. And so if you got on the Uber, I was saying, the guy says, "Oh, sure, I'm going to take you to Lehman." But at that point, because he was talking to you, even with the mask on, you can smell the guy clearly with alcohol. What do you do?
You exit the car.
Very good. Why do you exit the car? Yeah, you get out. Well, because the guy is going to kill. The guy the guy will probably get into an accident and you get hurt, right? Well, okay. So now you have I just put you more on alert when you Uber. Okay, so the bacteria, the Lambda virus, it gets into the host cell, and then it smells the bacteria. Say, is this a healthy bacteria, or this is a drunken, about to die bacteria? If it's a healthy bacteria, then it will say, okay, I'm catching right, and I'm going to get into your chromosome. And that's the lysogenic cycle. So healthy bacteria, unhealthy bacteria. Okay, so this is an exit, right? This is a ride. Okay, so now you understand how it chooses. Okay, and but you got into a situation, the guy who didn't talk to you and said, okay. You want to go to Lehman? He says, okay, all right. The guy didn't say anything, right? You just say, hey, take me to Lehman, and the guy knocked on the head and said, and so, but in the right, and you guys start driving. You didn't know the guy was drunk. So let's say what happens. So now you follow the lysogenic cycle, and this is where the regulatory protein binds. Okay, I'll come back to this very important moment here. And there's one protein actually binds, controls which side it goes. This is called the C1 protein. And so now I just said that you smell the guy. But how do bacteria smell? Well, bacteria can't smell, right? Because bacteria doesn't have a nose. So how does bacteria know if a bacteria, I mean, how does a phage know if a bacteria is healthy? And healthy bacteria can do what? And healthy bacteria will transcribe and translate things very fast, right? Because it's full of energy. It makes lots of proteins, right? So this C1 protein, so the speed of the translation determines the health of a bacteria. And so this C1 protein, the C1 gene is here. And if it's, so the C1 protein, it will be transcribed and translated. And then if the C1 protein is and the C1 protein will bind here first, site 1, and then site 2, and then site 3. Okay, so if a bacteria is healthy, that means the protein, this gets produced and stopped. Okay, and so you see this gets transcribed and the protein comes here and then stops. And this is the site that that will produce the protein to lyse and to lytic cycle. So this will be the lytic, and so lysis. All right, and so if the protein, so now if the translation is slow and this protein is made very slowly, then this promoter doesn't get blocked, and then you go lyse. And so this is how the virus can actually smell the host, whether it's a healthy bacteria or not healthy bacteria. Well, once it's and if it's a not healthy bacteria, then it will start reproducing. Right, it goes to the lysis, and so when it's lyse, what happens is that it will reproduce, and so when it reproduces, it goes to a rolling cycle replication. So this is similar to the conjugation that we talked about. So if you have a double-stranded host, and then what happens is this strand was here, right? Gets taken apart, and then it gets pulled that way, right? When this is pulled, and this has a 3' end, and that could be extended continuously. So this is almost like a toilet roll. Just think about a toilet paper. You just drag, and then it's rolling. And while it's rolling, it's replicating. So this always is double-stranded. And when it comes out, and then this part gets replicated. So this is what's called the rolling cycle replication. Okay. So that's how lambda DNA gets replicated, and the genome is actually once it's replicated, it has these and then I'm sorry, and so that's the replication. Now, and we know the rest of the story, right? Because that's going to the lytic cycle, and but if it smells the bacteria is healthy, then what's going to happen is the integration. So what will happen is the host chromosome. This is the lambda genome. It's inside the phage head, injects into the host cell, and it circularizes. And then it has this integration site, the attached site, from that has the corresponding site in the phage in the host chromosome. Okay, and then with one recombination event, the lambda gets integrated into the host. Okay, and so now it's integrated, and that's going through the life cycle, lytic cycle. Okay, so now I just wanted to highlight that you can have lytic cycle and the lysogenic cycle, and those are the key points. And so here again showing you the critical path. So this is integrating to. This is it smells good, integrates into the life cycle, and goes happily at this point. Goes happily, right? You're driving towards Lehman College. The guy who's and then in the middle of it, you ask the guy and said, "How are you doing?" It's just have a random conversation, right? And the guy turns around and says, "Oh yeah, I'm okay. I'm doing pretty well." And that's the point that you smell alcohol. So you're halfway through to Lehman College, and now you smell the guy and say, "Well, oh my God, he's drunk." What do? Again, at the next traffic light, whenever it stops, you jump out, right? Because you don't want to go down with the guy. And that's for bacteria, that's the induction. So bacteria could be healthy and growing happily ever after, but they're going to run out glucose, right, at some point. So if they run out of glucose, what would happen to the bacteria? They're going to die. And the phage says, uh-oh, this guy ran out of the energy, is going to die. And what happened? Well, it goes into the lytic. And so again, so how does bacteria actually know it ran out of the energy? So if the bacteria runs out the energy, what happened? If they don't have energy, can they translate? Can they transcribe? They can, but the transcription translation will be slowed down. And once it slows down, this site is not going to be blocked. Okay, so let me go back to here. And where is the directions that we were talking about? So yes, Mamadou.
Hey, Professor, I see lytic and the lydogenic. There is another difference, right? Because one cell is destroyed and the other cell is not destroyed. So is that another characteristic?
Yes.
Okay.
Yes. Okay. So this is where it takes place, and so the replication. Remember this slide. So I wanted to say CRO and C1, and then so this is where the CRO is and the C1 is. So it depends on which direction it goes. If when translation slows down, the energy level goes down, and this is not blocked. The promoter R is not blocked by operator O, and then this replication will start. Okay, so this will start replication. So when low energy comes, or bacteria goes into starvation, or it slows down, then what happens is that bacteria actually goes into the lytic cycle, and also on this side. So when C1 protein slows down, and the repressor, there's not enough repressor to keep the operator occupied. So both CPR and PLF will become active, and you have lambda being made, and the lambda exit protein gets made. Okay, so that's the life cycle of the lysogenic bacteria. Okay, so those are, I spend most of the time talking about the bacteria virus as an example. I do want to give you some introductory basic information about vertebrae viruses, which we won't be able to spend most time on. And so in terms of the structure, it varies a lot, and you have different types of virus. And you might have heard about adenovirus. Anyone heard about adenovirus? No? How does Johnson & Johnson makes their vaccine? Okay, that's a question for you to look up. Yes, Sean, go ahead.
Don't they use chimpanzee adenovirus to like carry the protein particles that the cell will recognize are viral particles and destroy? Right?
Yes, that's your vaccine. Johnson & Johnson uses this adenovirus as the carrier for vaccine. Okay, and the protein inject, and that's how they make it. Anyone heard about herpes virus? No? I thought it was popular, and no? I heard that love is not forever, but herpes is forever. Never heard this? Yes. Yes, herpes viruses. That's what they look like. Is a double-stranded DNA has an envelope, and once you have them, it stays with you forever. There are other interesting viruses, and with different viral particles, enveloped and non-enveloped. And here, again, the genome varies a lot. It could be anyone. And so here I wanted to highlight why is it possible for virus to have different genome, right? And what happens when they have different genome? Well, doesn't matter what they have as a genome, they all have to have mRNA. Okay, so they all have to produce mRNA in order to carry out translation. And so the genome to mRNA will require different steps. Okay, and so this is where it's important for you to understand that if you have the double-stranded DNA, well, that's straight, and then you transcribe, then you have mRNA. And the mRNA, in this case, is defined as the plus strand. Okay, this is important because the mRNA could be single, well, you can have single-strand RNA, right? Then you worry about the plus or the minus, which one is being translated. The one that's being translated is the mRNA. So if you have single-stranded plus virus, then you have to get a double-strand DNA in order to have what? In order to have an SS, you have to have a single-stranded DNA, but that's a minus strand, right? Because minus strand is the template for the plus strand mRNA. Remember the anti-parallel? This is where you need to be very clear. The mRNA is the plus strand, and then you're going to need to have a minus strand in order to do the transcription. So if you look at the double-stranded RNA virus, and that's straight, and you need to transcribe the minus strand. And if you have a minus RNA, and that needs to be transcribed. But if you have you have a single-stranded plus strand, that's already mRNA. Okay, and that's directly used as the mRNA. If you have a single-stranded retrovirus, yes, you're right. Go ahead with your question.
So for the plus strand, it uses a minus strand as a template. Is that how it is?
No, yes. So if the genome is a plus strand, right? And so I'm here. Wait, where is my? Okay, so let's take this as an example. If the plus strand is our RNA, and that equals mRNA that could be used directly. However, if you want to reproduce this single-stranded mRNA, and that needs to do what? You have to go to, if you have to make this, then you have to make a RNA minus strand, right? And then you can reproduce. Wait here. Then you have an A plus, right? So you go from, and so yes, you can, for this particular virus, it's easy to see how fast they can be translated. But in order to reproduce itself, it needs to make a template of the complementary strand and then make copies of it. Is that clear? Same principle applies to everything else. Okay, thank you. The effect of the virus in eukaryotic virus is very, very complex, and they could do multiple things. Different virus does different things, and so some virus will transform and cause the whole cell to form. These are form tumors, and some kills, and some have persistent infection, meaning they can continuously spill out the virus, but the cell is still alive. In this case, they tend to form a bud because the eukaryotic cell has membrane, it's flexible, and they can have like a drop, which will later on will show, right? And they can have self-infusion, they can have they can also have latent infection. And the latent infection is, in this case, the virus is there, just like lysogenic cycle. It's being carried, but there's a trigger point, and when there's some event, will trigger gets them into the lysogenic cycle. All right, and you can also have phage plaques, and that's how they count the animal virus, these white spot. And I'll take one particular virus as an example. The retrovirus. And you probably have seen this picture in many different classes, but this is the typical retrovirus. And in terms of structure, it has two copies. It has two copies of the RNA genome, and it also carries enzymes. Reverse transcriptase and integrase and protease. Okay. Now, does bacteria carry any of? Does bacteriophage carry any of the enzymes? No, the bacteriophage doesn't. Why? Well, because it uses everything from bacteria cell. And retrovirus or adenovirus or any other virus doesn't have to carry enzymes, except this retrovirus carries the enzyme, the reverse transcriptase. Well, we know the function of reverse transcriptase, right? Do we? Well, we know transcription goes from DNA to RNA, and then if you have to reverse, it's reverse transcriptase, goes from RNA to make DNA. And then you have integrase. Integrase, the DNA, the DNA will actually get into the integrates into the host chromosome. And the protease is the enzyme that actually will process the proteins into the correct form. And you've probably heard about protease inhibitors as the HIV or AIDS treatment. Have you heard about that? And so that's the protease. So if you inhibit that, and the HIV virus will not be able to produce a functional viral particle. Then it has lipid membrane, and membrane comes from the host chromosome, host cell. Then it has a core protein, shell protein, and then it has core protein. And so all of these are encoded by three types of three sets of genes, in fact. Okay, so this is the genome. Okay, so that's what the mRNA is. So if we were to look at the life cycle of the HIV, you would see that it has the viral particle. It has two, and it gets entering. Now, this entering part is very important. This is where it's different from bacteria and the eukaryote, from phage to virus. Phage only injects its DNA into the host cell. The retrovirus, however, just because it's a membrane, and it's like membrane drop, it's like the oil drop. So if you have one oil drop, a small tiny oil drop, and then you put it to the next oil drop, two oil drop will fuse together, right? So imagine one virus is a drop of oil, and your cell is a giant drop of oil. When the two meet together, they fuse. And once they fuse and it gets uncoated, the part is there. These core protein all gets encoded, digested, and then the RNA gets released, and that's where the reverse transcription takes place and makes double-stranded DNA, and the double-stranded DNA carries the integrase and goes into the host chromosome and integrates into the host chromosome. And this is where the part that's really a problem. Because once it gets into the chromosome, there is no way to precisely cut it out. So the only hope is that you hope this host cell will die along with the virus. That's where the problem is. And the challenge is curing the HIV infection. And so once it's in the genome and then it gets transcribed just like the regular chromosome, any other genes, and transcribed, translated, assembled a new viral particle. So an infected cell will continuously produce HIV viral particle. And the treatment always is hoping that you block somewhere here. You reduce or you block, you treat this cell so that it doesn't produce a functional HIV virus. So, for example, you can use analogs or protease inhibitors so that the protein that's produced, the protein produced is not, or they don't produce enough protein, or that the protein they produce is not fully functional, or the RNA, it gets so much mutation on it, the genes in the new viral particle has so much mutations, the protein that it produces is completely wrong and has no function. Okay, so those are all the points that we're trying to attack and inhibit the virus from reproducing. Okay, so that's an example of vertebral virus, and we picked the one that's we know most, not we know most, but it has the most significance to our life. And lately, in the last 18, 20 months, and this virus has been causing major problems, all right? And so COVID-19 virus, this is the structure. It has the spike protein on the surface and the genes. And so now what we are, the Pfizer and all the Moderna virus, the vaccine, is that we get a nanoparticle, we get a piece of the gene or the mRNA that codes the S protein. So now the S protein, so this the viral, the nanoparticle gets into our cell produces the S protein, and then to be recognized by our hosts, our immune system, so that we generate antibodies against the S protein. So when the real COVID-19 comes in, we can have defense, IgG against that part. Now I will come back and spend more time talk about the COVID-19, because to understand how it works, we need to understand our immune system as well. So that's why I wanted to say this particular slide, even though this virus is very, very important, it's on top of our mind for the longest time, well, the last 20 months, we will, this discussion will come for exam 3, will not be on exam 2. Okay. Okay, so that's the end of today's lecture. I'm gonna stop here, and I'm first gonna stop the